Welcome to My Puppet Health. Today we are discussing how to measure your biological age. Biological age refers to how old a person's body appears based on various physiological markers, as opposed to their chronological age, which is the number of years since birth. Advances in science have allowed for more precise measurements of biological age, one of the most accurate being DNA methylation testing. This essay explores the different methods of measuring biological age, the science behind DNA methylation, and how it was used to determine that my biological age is 11 years younger than my calendar age. The science behind DNA methylation. DNA methylation is an epigenetic mechanism where methyl groups are added to the cytosine nucleotide of DNA, typically at CPG sites, regions of DNA where a cytosine nucleotide is followed by a guanine nucleotide. This process can activate or repress gene expression without altering the DNA sequence. Over time, the pattern of DNA methylation changes, reflecting an individual's biological age. The most accurate method for assessing biological age through DNA methylation was developed by Steve Horvath, known as the Horvath Clock. This method uses specific CPG sites as markers to estimate biological age with remarkable precision. The Horvath Clock analyzes DNA methylation patterns from various tissues, providing a comprehensive age estimate. In my case, DNA methylation testing revealed that my biological age is 48.6 years, while my calendar age is 59.6 years. This 11-year difference indicates that my body's physiological condition is younger than my chronological age. Components analyzed in DNA methylation testing. To calculate biological age, various markers are analyzed and compared to population averages. Here are the specific components included in my DNA methylation test. Albumin. Albumin is a protein produced by the liver, essential for filtering and distributing nutrients. Low levels can indicate liver or kidney dysfunction or protein deficiency. Normal albumin levels suggest efficient nutrient distribution and waste removal. Creatinine. Creatinine is a waste product filtered by the kidneys. Measuring creatinine levels helps assess kidney function. Normal levels indicate healthy kidney function, while abnormalities can signal potential kidney issues. Glucose. Blood glucose levels are crucial for diagnosing insulin resistance and diabetes. Consistently abnormal glucose levels can accelerate aging and increase the risk of age-related diseases like heart disease and dementia. High-sensitivity C-reactive protein, hsCRP. hsCRP is an inflammatory marker. High levels indicate chronic inflammation, a hallmark of accelerated aging. Low hsCRP levels suggest lower inflammation and a potentially slower aging process. Lymphocyte percent. Lymphocytes are a type of white blood cell crucial for immune defense. Abnormal lymphocyte levels can weaken the immune system, increasing susceptibility to illness. Normal levels indicate robust immune function. Mean cell volume, MCV. MCV measures the average size of red blood cells, important for oxygen transport. Abnormal MCV levels can indicate nutritional deficiencies or blood disorders, impacting overall wellness. Red blood cell distribution, width, RDW. RDW measures the variation in red blood cell size. High RDW levels can signal cardiovascular issues and other age-related conditions. Alkaline phosphatase, ALP. ALP is an enzyme found in the liver, bones, and other tissues. Abnormal ALP levels can indicate liver, bone, or bile duct issues, affecting biological age. Advantages: highly accurate, provides detailed molecular data, reflects cumulative biological aging. Disadvantages: more expensive, requires specialized equipment and expertise. Other methods of measuring biological age. There are several methods to measure biological age, each with varying degrees of accuracy and detail. Telomere length. Telomeres are protective caps at the ends of chromosomes that shorten with each cell division. Measuring telomere length can provide an estimate of cellular aging. Here's how this process works in more detail. Sample collection. Blood samples are typically used, though telomere length can also be measured in cells from saliva, skin, or other tissues. DNA extraction. DNA is extracted from the collected cells using chemical or mechanical methods. Telomere length measurement has limitations due to variability in telomere length among different cells within the same individual and across individuals. Factors such as genetic differences, stress, and lifestyle can also influence telomere length, making it a less reliable sole indicator of biological age. Advantages: provides a direct measure of cellular aging, relatively simple to understand. Disadvantages: high variability influenced by factors such as stress and lifestyle, not as accurate for determining overall biological age. Biomarker analysis. Biomarker analysis involves assessing levels of specific biomarkers in the blood that correlate with aging. Biomarkers include proteins, hormones, and other molecules that change with age. The process involves several steps. Sample collection. Blood samples are the most common source, although urine, saliva, and other bodily fluids can also be used. Biomarker selection. Common biomarkers related to aging include proteins, such as albumin and C-reactive protein, CRP. Hormones, such as cortisol and insulin. Metabolites, such as glucose and cholesterol. While biomarker analysis is informative, it can be less precise due to the influence of external factors like diet, lifestyle, stress, and medications. Multiple biomarkers are often analyzed together to provide a more comprehensive picture of biological aging. Advantages: can measure a wide range of physiological markers, useful for tracking changes over time. Disadvantages: biomarkers can be influenced by external factors, requires multiple tests for comprehensive assessment. Functional age tests. Functional age tests evaluate physical and cognitive functions to assess how well an individual is aging. These tests provide practical measures of aging but are less specific at the cellular or molecular level. Here's how they are typically conducted. Physical function tests. Grip strength, measured using a hand grip dynamometer to assess muscle strength. Decreased grip strength is associated with increased mortality and disability. Walking speed. Timed walking tests, such as the 6-minute walk test, measure mobility and endurance. Slower walking speeds correlate with higher biological age. Balance tests assess static and dynamic balance, which can indicate neuromuscular function and fall risk. Cognitive function tests, memory tests, evaluates short-term and long-term memory through tasks such as word recall. Executive function tests assesses higher-order cognitive processes, including problem-solving, planning, and task flexibility. Common tests include the Stroop test and the trail-making test. Processing speed tests measures how quickly an individual can process information, typically through timed tasks. Advantages: provides practical insights into physical and cognitive aging, easy to perform. Disadvantages: subjective and less precise, does not provide molecular-level data. DNA methylation is considered the most accurate method for measuring biological age due to its ability to capture the cumulative effects of aging at a molecular level. Unlike telomere length or biomarker analysis, DNA methylation provides a direct readout of gene regulation changes associated with aging. The precision of the Horvath clock and similar epigenetic clocks allows for highly accurate biological age estimates, making it a powerful tool for assessing overall health and aging. Understanding biological age and the methods to measure it is essential for proactive health management. DNA methylation testing, due to its accuracy and detailed molecular insights, stands out as the most reliable method for determining biological age. My test results, which show a biological age of 48.6 years compared to a calendar age of 59.6 years, demonstrate the potential of this technology to provide meaningful insights into one's health. By regularly assessing and managing biological age through targeted interventions, it is possible to maintain better health and potentially extend lifespan. On my website, mypuppyhealth.com, I have an at-home biological age test. You do not have to provide a blood sample. This test collects DNA samples from inside your mouth with a swab. In addition, I also offer other blood marker tests that measure various biomarkers, including nutrients like vitamins and minerals, proteins, enzymes, and metabolites. This will allow you to select the supplements that can correct any of these markers that are out of range. I have also sourced a USA manufactured line of supplements. All products are third-party tested to ensure the quality of the ingredients and to make sure no heavy metals have made their way into the formulations. Visit my store at mypuppethealth.com, enter the code PUPPET and receive a special discount. Make sure to like, share, and subscribe for more health videos on my channel.